How many passages are acceptable for mesenchymal stem cell treatments? Hi, I'm Josh with Dream Body Clinic, and that's what we're going to get into today. This is a question that I get asked all the time, and the answer is simple. We typically will go to the fifth or sixth passage, but it's not as simple as that. You have to understand what a passage is, where it's going, and why we do it. So let's start at the very beginning. We work with a group of gynecologists that only work with first-time mothers. When you're doing mesenchymal stem cells and you're going to replicate the cells, you want the youngest, healthiest source. So we have to be very selective in who we ask to donate this tissue. We want first-time mothers. We want them to be between the ages of 18 and 25. We want to know how many sexual partners. If they answer more than one, disqualified. Because an MSC can't carry a virus, but if the donor had a retrovirus, there's a less than 5% chance of crossover. Most of those are sexually transmitted, so we're vastly narrowing down this funnel for the best tissue. We ask if they've had any of the COVID vaccines. If they have, disqualified. There's a federal database that everyone who took it was registered with, so we double check. So very young, healthy cells. The women are thoroughly screened. We don't just take their word for everything. We check, we verify, and make sure that they have no retroviruses or any other issues. We want to make sure they're healthy. Then, when they give birth, we have about 6 hours to take that umbilical cord and placenta from the birthing room to the lab. There's a whole sterilization process there, antibiotics, and then we have to start with the culturing process. And in the lab, we take it there and you start dissecting it. It's pretty gross. Looks like little pieces of meat. They're putting on petri dishes, and then they're taking that into these beakers that they're filling up with collagenase and that tissue, and they centrifuge it. The centrifugation process separates the cells from the tissue, and then we go further and we start with shaker tables, which shakes the cells out of the tissue. So now we're left with this liquid. And we then take that liquid and fill the flasks. Now, we don't fill them all the way. There's measurements of how much that they put in. And then we add the culturing medium. The culturing medium is the food for the stem cells. So they eat through that, and we put them into the incubator, which is like a specialized oven with exact CO2, exact oxygen levels. It's a biological oven, essentially. So you're keeping it the perfect temperature with all the food they need. It's like the Garden of Eden for mesenchymal stem cells. They start replicating, but in the replication process, they're growing, they're eating, they're consuming that culturing medium. So we get to a point, which usually the first passage will take anywhere from like a week to 2, and then we're able to take those out, and we have to do what's called a flush. You have to flush out the other types of cells. You're not left with just pure mesenchymal stem cells. In fact, a third or more are going to be mostly red blood cells. The placenta doesn't really carry any hematopoietic stem cells, but the Wharton jelly does, so you've got to flush that out. Those are the stem cells that make red and white blood cells. And you've got to get rid of monocytes, you've got to get rid of all these other cells, because those other cells have the donor's HLA. So if they were put into a person, they wouldn't really hurt them, but they would just be eliminated by the immune system. It's not like getting a donor organ, where it would continue to attack and you would develop what's called graft versus host disease. With these sort of treatments, there's no risk of that, because even if there was differentiation, meaning a MSC turned into a chondrocyte, an adipocyte, or other cell, it would exhibit the donor's HLA, but your immune system would wipe it out. Fortunately, MSCs don't exhibit HLA, so we're not worried there. And this is a good point to make with what they do in the United States, because they're not allowed to do this flushing process. They keep that third or more of other types of cells in what they give you. If you're getting cells from a doctor in the US, these frozen umbilical cord cells, a third or more are actually other types of cells that will be rejected by your immune system. And in that process, they tend to kill a lot of the mesenchymal stem cells in there too. So when they're telling you you're getting like 10, 20 million cells, well, know that it's much fewer than that, and the freezing process can kill them, plus a lot get killed by the immune system because of all these other cells in there. It's not a very good way to do it. They should at least let them do a flushing process and get pure, because the flushing process, so let's get back to the flask, culturing medium, they just ate through all that culturing medium, their food's gone, they need more food. We flush, we put in more culturing medium. The mesenchymal stem cells don't get flushed out because we use a specially type of plastic in these flasks that they adhere to. So they stick to that, the other cells get washed out, and you're left with pure MSCs. You put in more culturing medium, they then have food again, they keep growing. So each time you do that is considered a passage. So you do not have pure isolated MSCs until at least the third passage. There's a lot of other cells you got to get rid of. And if you look at like the International Stem Cell Association or other guidelines, like in any study that's done with cultured stem cells, they will not even begin that process until they're at least to the third passage. So I'm hearing a lot of these clinics out there, oh, we only go to the third passage, or that's what they're telling potential patients. And it's ridiculous because that's usually when they start counting. So if they're telling you they're going to 3, they're probably going to the 6th. They're going 3 more, which is totally fine. Like you can go up to the 7th passage with no issues at all. By the 10th passage, there are studies that have proven and shown that you start having deterioration of the cells. They do not evolve and get better, they tend to devolve. They start getting oblong, they start getting bigger. You have different issues. They still will work the same way, and technically you could keep culturing, to the 100th passage if you wanted to. You're just going to have a lot of weird, misshapen cells, and some of them will even try to differentiate if you go too far. So at the 10th passage, you never want to go there or further. The 12th passage, it is guaranteed the majority are going to have those deformities. They're not usable. So the whole passage thing's ridiculous. They're going to try and tell you, oh, they get weaker, they're worse the more you replicate them. And that does happen, but not till like around 10, passage 10. So you have to go a long ways. You would have so many cells at that point, you'd have to start freezing them unless you had like hundreds of patients per day. It's just not even possible. We treat around 150 patients per month, and we're using one umbilical cord maybe every month or 2, maybe 3 months. It just depends on patient load and how many cells each patient is doing. So it's not like we're going through a lot of this, and we want to use it so that it's never frozen. So we take them straight from the incubators, we bottle them up for treatments, and then we always administer within 24 hours because we don't want to use frozen cells. So we have different cultures in the incubators. There's racks so that we can have different cultures and different flasks always coming out daily so that patients get the best, freshest cells. You don't want frozen cells. Just the freezing process alone can kill the majority of them, and the thawing process, if not done properly, will kill most, if not all. It's just not a good option, and they're not teaching a lot of doctors how to use the different ice baths at different temperature grades to thaw them properly. Now in research, that's all they use is frozen cells. So it is possible. You just have to do a whole process, and you're still going to have some die-off and issues. So the whole idea of like this marketing game of how many passages is pretty ridiculous. Nobody out there is going far enough to have any issues, to have weaker cells. What really matters is that you have a clinic and a lab like we have that does it the right way, that uses the best materials. It isn't using bovine serum, which a lot of these are. Pretty much all of the labs in Mexico other than us, that's what they use. It's for animal use. You don't want that. You want xeno-free mediums. You want them for human use. You want to make sure that you're getting the best quality so that you can have the best cells with the best results, which we continue to prove. I mean, if you can see, I'm probably the only founder of a clinic out there that's doing treatments on itself. I've done the intrathecal treatment twice. It's into my spinal cord fluid, goes to the brain. Like, if there's one treatment people think, oh, that could be dangerous or scary, that's the one. It's super safe if you're using good pure cells like we do. My wife's done it, my mom's done it, my father-in-law just did it 2 weeks ago. We believe in what we do. You can see I do the IV and facial about every 6 months. So does my wife. My kids have done IVs. We believe in what we do, I guess is the point that I'm trying to make. We know how far to go. We know what to do in the lab to make sure we're giving the best cells possible, the best results. There's a lot of people out there just saying ridiculous things. There's some of these US doctors that are just out there thinking, oh, if they bash like cultured stem cells, it'll make people want to go to them. It's like, explain why your cells are better. You don't have to say bad things about anyone else. I've never seen that help anyone get more business. I've never seen these other labs that will try to make these little distinctions. Oh, we only go to this passage or that. Like, these are new points, and nobody's going to a passage of ground that is far enough to have any issues, because nobody wants patients to have bad outcomes. They want them to have good outcomes. So it comes down to that. You got to be able to make the right decision and get the right info. So we'll keep giving you the info. If you've got questions, if you want to learn more, that's why I give everyone my phone number. It's on the website. You can call. Either my assistants will pick up and get you on a call with me, or you can call me direct. I'll answer your questions, make sure you understand. We have some of the best doctors and biologists in the world working for us. They're happy to answer questions too. I'm American. Most of our patients are American, Canadian, UK, Australia, these English-speaking places. I understand this, I can explain it to you, but we rely on the doctors and biologists. They are the experts, they are doing that. I'm just here to communicate that to you. So I'm here to help. I'm here to answer your questions. So is our whole team. We've Dream Body Clinic. We've got well over 40 people that work here to help make sure that you get the best quality stem cells at the best price in the best place in tropical paradise here in Puerto Vallarta. It's pretty awesome. So if you want to learn more, get a hold of us anytime. Call toll free at 833-445-9089. Check out our website. It's got all the treatments listed on the homepage. You can scroll down there at www.dreambody.clinic. All the prices and info are there. We go out of our way to be as transparent as possible. Everything's there for you to make a good, informed decision, because there's a lot of great options out there. We're hoping you realize that we're the best, but do what's right for you, and we'll go from there. So I'm Josh, happy to help.